Bioengineering seminar series, we have Dr. Danny Bluestein from Stony Brook University. He received his PhD in biomedical engineering from Tel Aviv University in Israel. He is interested in how physical forces regulate cellular function and blood flow, and the translation of this knowledge to numerical and experimental strategies to improve the design of blood recirculating devices, including prosthetic heart valves, ventricular assist devices, and artificial hearts. He has over 140 peer-reviewed scientific articles. He has served as the associate editor of ASME Journal of Medical Devices. He's a member of several different boards and societies. He has funding from the NIH. He is a fellow of the American Institute of Medical and Biological Engineering. He's also a fellow in the a fellow of the Biomedical Engineering Society. And he told us today, and he has on his presentation slide that you can see, that he is the winner of the 2021 Salvio L. Wu Translational Biomechanics Medal Award from ASME. This is a huge, huge honor and a fantastic award that really highlights researchers who take our engineering and our research and can actually bring it to the clinic and help patients. So that's just a fantastic award, and I'm so excited for Danny. His presentation today is going to be the optimization of prosthetic devices and procedures for treating cardiovascular diseases and the biomechanics of thrombosis across the scales. So with that, I'll turn the floor over to him, and you can begin your presentation. Thank you.
Thank you for this generous introduction, and I wish that I could actually arrive in person to your beautiful campus, but I got it that we need to wait. I was actually scheduled to arrive in April, and necessarily this was postponed, so I'm very happy for the opportunity to do the same in Zoom. So let's just embark on this. It's going to be a pretty busy ride. Okay, so when we're discussing optimization of mechanical circulatory support devices, what we would like to do is minimize the thrombogenic potential or thrombogenicity, or basically enhance the thromboresistance. And why is that? First of all, congestive heart failure that may necessitate the use of such devices has a high prevalence, around 5 million patients in the US alone, 15 million worldwide, and over 500,000 new cases are diagnosed each year in the US alone. Now, when we're discussing such devices, we may actually look at the prosthetic heart valves, like you can see here, ventricular assist devices, which are pretty crazy type of devices, or even the total artificial heart. Some of you may not realize that there are more than 2,000 patients that are implanted with the total artificial heart that are walking around with us. Now, the problem with all these devices is thrombotic and thromboembolic complications, which is a major impediment to such devices. They are burdened by formation of blood clots and thrombus formation that mandate complex pharmacological lifelong anticoagulation and anti-thrombotic therapy. Now, in order to do that, the traditional practice of research and development of such devices was more kind of trial and error and established experience. Now, we set out to develop a predictive technology that has the potential to minimize device flow-induced thrombogenicity. And there's a good reason we focus on flow-induced thrombogenicity. Now, the general title of this methodology is device thrombogenicity emulation, or DTE, which is a kind of a universal methodology. So I'm going to walk you through the steps. Now, for many years, computational fluid dynamics, or CFD, is used for developing and designing such devices, but it's still focused on improved hemo or hydrodynamics, which is anticipated to reduce the thrombogenic potential, but it's not directly correlated to, for example, coagulation markers of a blood clotting that generates this. The DT methodology, the idea is to optimize the thrombogenic performance by testing virtual device design modifications in silico before the prototypes are built and tested in costly and preclinical trials. Now, why blood flow? Non-physiological flow patterns enhance the hemostatic response of blood, and mostly by chronically activating platelets, which is the preeminent cell that initiates the coagulation cascade. Now, platelets react to either biochemical agonists or mechanical stimuli by a dose and time response. In terms of pathological flow stresses that characterize such devices, it translates into stress accumulation that eventually may drive the platelets beyond the activation threshold, and then the coagulation cascade starts. And this is exactly the reason why the patients require lifelong anti-coagulation. And the question is, can we optimize the devices during the R&D stage to minimize this thrombogenicity? Now, first of all, I would like to describe you a little bit the response of the platelets to elevated shear stresses. So suppose that we have here a quiescent platelet flowing in the blood flow, exposed to a velocity gradient that generates shear stress. Upon activation, and this happens very quickly, the platelets completely change their morphology. They express anionic phospholipids, start secreting clotting factors like factor 5A, 10A, and also mediated by fibrinogen from the blood plasma and from von Willebrand factor start aggregating, and they provide a kind of enlarged surface for reactions of something that is called the prothrombinase complex. And what the prothrombinase complex does is convert prothrombin into thrombin, which is a very strong agonist that acts in both ways. It immediately feeds back to reactivate the platelets and accelerate the process, because the idea of activated platelets is block blood spillage. And they also convert soluble fibrinogen from the blood plasma to fibrin, which is the actual clot material. Now, thrombin, this preeminent factor that is responsible for this conversion, is a direct result of the activation of the platelets. Now, if we would like to measure that, and in the DT methodology, this is very important that besides numerical simulations, we'll be able to validate our results with some assay. We have here a problem because the thrombin feeds back to reactivate the platelets, which is a product of the platelet activation. And the question is, how can we determine what was the contribution of the platelets? So quite a few years ago, with a hematologist, who's a non-thrombosis expert, Julian Jesti, him and me developed this platelet activity state assay, which mimics kind of congenital defects of prothrombin, prothrombin quick 1 and METS prothrombin, which is kind of similar to hemophilia kind of syndrome. And by modifying the prothrombin, by acetylating it, we generated a species of prothrombin that can still interact with the prothrombinase complex to generate defective thrombin that doesn't feed back to reactivate the platelets, nor does it clot the fibrinogen into fibrin. This is actually very important for in vitro experiments, because if you block this, you don't have a formation of blood clots that may totally block your recirculation flow loop. So this is how we measure platelet activation that is flow induced, and with additional benefits that this could be done in real time, unlike flow cytometry, for example. Now, regarding numerical simulations, this is something that we've done already a decade ago or so. And this is a fluid structure interaction of a mechanical hard valve during the opening and closing. And what you see here is a dispersion of a cloud of platelets, 10,000 of such, during forward flow over here, and here at the bottom, you can see this during backward flow or during the diastolic phase of the heart pumping. So this is actually the point of view from the left ventricle. Now, we compared here in the numerical simulations two bileaflet mechanical heart valves that were very similar in design. Actually, the two companies sued each other about the design. The only difference is the hinge mechanism of each of the valves right here. And what you can see that this has a major effect on the reverse flow going backwards through the closed valve in terms of the velocities, the kind of micro jets that are generated here that are very activating to platelets. Now, in order to estimate what it does by these numerical simulations, we conducted the fluid structure interaction modeling and using two-phase flow of blood plus platelets. And then we computed for each individual platelet its stress accumulation, which is basically the dose time response of platelets and its accumulation. So this is done by the product of the shear stress and the exposure time to it and summation along numerical nodes. Now, when we say something like that, to look specifically how we do that, each platelet flows along its own individual flow trajectory. And as you can see, those could be very complex flow patterns. So for each such platelet trajectory, we then have a loading waveform in time, and you can see very rapid instantaneous changes in the stress level along the trajectory. It keeps changing all the time. Now, then we extracted from numerical simulations from the total stress tensor scalar for each location of the platelet. This is actually kind of similar to von Mises stress, if you are familiar with. And then we computed the stress accumulation. Now, if you do that for like 50 or something, 80,000 such trajectories, it generates a huge amount of information. So we then, basically what we were interested in each individual trajectory, what was the level of stress accumulation that the platelet achieved by that. Let's say around this point in the wake of the valve, where it actually may have reached the threshold for activation, and there are certain established threshold for this dose time response. So to collapse this huge amount of information into something that we could actually look at and interpret, we did probability density function of the distribution of these values of the stress accumulation in, by the end of each platelet trajectory. And we termed this the thrombogenic footprint of the device. So these two bars that we compared earlier, you can see this type of distribution. Now you see here a very long tail, and this is actually the risk zone where platelets may get activated. And the general mode of the distribution is such that it's much lower, way below the threshold for activation. But if you see differences right here when you zoom in the tail region, this would tell you, okay, this valve is far more activating than the other valve. So it informs you about the right design. Maybe, for example, the hinges area. By the way, it's a good medical back then, bought this company 8 years and basically used this hinge design exactly for that reason. Okay, now let's move to more complex devices like left ventricular assist devices. I mentioned earlier that those are crazy devices because they spin at around 10,000 RPM. Now, basically, when we have a failing heart, we connect the bottom apex of the left ventricle to this left ventricular assist device, and it kind of sucks the blood from the ventricle and propels it with a huge momentum, and then introduces it back to the ascending aorta right here, bypassing the failing heart. Now, the only reason that the blood doesn't completely clots by passing through this huge elevated shear stresses that you find in these devices is that the exposure time is very, very short. And this is what saves the blood from clotting. Yet, you still mandated to use blood anticoagulants, which are very problematic to manage for patients. Now, when we look at what may increase or decrease the flow-induced shear stresses that may activate the platelets, it's actually necessarily design modifications within the device. So you can see that this device is very similar to a turbojet or a turbofan jet. It has an impeller, it has a stator, rotor, and there are many ways and many parameters in the design that you can kind of introduce and optimize it. But the question is how to go about it and which parameters would necessarily reduce these shear stresses. So in order to quantify that, and here just in general, this is more like design indications for ventricular assist devices. So we collaborated with this company, Micromed, in optimizing the design from what was originally called the Baker ventricular assist device to the optimized Heart Assist 5. And I'll describe to you a little bit the DTE, device thrombogenicity emulation methodology. In the heart of it, we have a programmable hemodynamic shearing device. And this is a combination of couette and cone plane device that generates a uniform shear stress while the cone is spinning and generating the shear stress, and we can expose blood samples to it and measure the platelet activity. with the platelet activity state assay or pass assay that I've just described. Now, the thing is that this programmable hemodynamic Turing device can spin in very different RPMs and change instantaneously according to loading waveforms. The loading waveforms that we are testing are something that we call hotspot trajectories that are derived from the numerical simulations. Here, for example, you see such 4 hotspot trajectories in which the instantaneous shear stresses were highest when blood was flowing through the device, and you can see them right here. And then we start reiterating the design, and then for each design iteration, we compute a new probability density function or the thrombogenic footprint that they have just described. And the idea is to minimize the risky tail zone that you see here and push the main mode to here, the lower risk zone. So we keep reiterating that, and after going through all these design iterations, all done in silicon. It's not like each time we build a new prototype and test it, but after enough such iterations, you can see that with all the design modifications, this was the original the Bakey device. This was the optimized design, far more streamlined if you can look at it qualitatively, less shear stresses. And the resultant instantaneous hotspot trajectories were really minimized to a very large degree. So then we freeze the design and tell the device manufacturer, build the prototype, and then we actually tested them. So this was the optimization. And then we took the prototype that the company built and tested in a small recirculation blood flow, but under the same physiological conditions that are functioning. And then we measured the platelet activity state, and mostly we were interested in the platelet activity rate or the slope of, here you can see, of the 30 minutes recirculation time. And this was the slope before optimization and after optimization. And we actually reduced the thrombogenic potential by close to 90%. Now, we were interested because the traditional approach to it is pharmacological, to compare the design optimization efficiency to anticoagulation effect. For example, here, compared to the effect of aspirin. Aspirin is a direct antiplatelet drug, or a more advanced antithrombotic anticoagulant called dipridamole. In each case, the pharmacological management could reduce at the most, in the case of dipridamole, close to 30%, while the optimization achieved a much higher efficiency by reducing to 10 to 15% of the thrombogenic potential. Now, this was actually used by the company during the FDA submission. Now, here you can see also results of such simulation comparing the optimized heart assist 5 to the most commonly implanted ventricular assist device. Back then it was called the Thoratec HeartMate 2. It was then purchased by St. Jude. St. Jude was then purchased by Medtronic, so nowadays it's implanted and marketed by Medtronic. And you can see that the optimized Heart Assist 5 actually has better thrombo resistance compared to the Heart Assist 8, to the HeartMate 2. Now, we also collaborated with Syncardia, the only FDA total artificial heart approved right there. You need to realize that the patients implanted with this device are heartless. That means that their heart is actually removed from their body and replaced with this mechanical pump. And no one would like to be in that position, but some people save their lives. So the problem is that the original design was pretty big. It had 70 cc kind of ejection volume for cardiac output. So it didn't fit small patients and many female patients in bed, and it wasn't good for pediatric use as well. So the company wanted us to test using this in silico in vitro DT methodology to see if they can reduce the size of the device to 50 cc and also for 35 cc. So full duty cycle fluid structure interaction and using the same methodology of setting it with platelets and quantifying the thrombogenic potential, generated the thrombogenic footprints, and saw that for all sizes, when we make the device smaller, although it increases the thrombogenic potential, it is still within the safe zone for minimal activation of the platelets. Eventually, the company Syncardia manufactured the 50 cc total artificial heart. It didn't go all the way to 35 cc, but the 50 cc has been implanted successfully in patients for the last 3 to 4 years, mostly in Europe. Okay, now I'm moving to a different type of device, the transcatheter aortic valve replacement. Those of you who are not familiar with it, I'll describe the procedure, but basically this is a prosthetic heart valve that is implanted without surgery, which is quite surprising how you could do that. And this is a kind of life-saving solution for severe aortic stenosis patients. Those are patients who have severe calcific aortic valve disease, which is a progressive disease with many calcifications to the aortic valve. And in the final stages, the heart valve hardly opens and closes, and the systolic blood flow increases a lot because of jet flow, very elevated shear stresses, the risk of thrombosis and cardioembolic stroke, etc. Left ventricle of the heart, the pumping chamber needs to work much harder. Severe aortic stenosis, risk of myocardial infarction, and eventually sudden death. And in that, in the severe cases, the survival rate in 4 years is very low. So for such patients, they also cannot tolerate cardiothoracic surgery. So there was an unmet clinical need to this high to intermediate, and by the way, by now also transcatheter aortic valve replacement is offered to low-risk patients. It's a minimally invasive intervention, but you can see here, for example, the most popular such device, the Edwards Life Sciences Sapien 3, the most famous patient in the world that received one of the first such tablets is Mick Jagger, jumping jack flash, and by the way, very recently also Arnold Schwarzenegger. Now, how this procedure works? The valve is crimped into a stent and delivered through the femoral artery, navigated through the aorta, and then, without removing the disease valve, it's deployed over the calcified valve, pushes it to fully open, and then you have a kind of functional valve. And as you can see, it anchors to place. It's not sutured, so there's no need for opening the chest and performing a full cardiothoracic surgery. Now, this is pretty problematic. There are many risks and complications that are involved. Now, the current technology uses bioprosthetic or tissue-based valves. Now, this is not live tissue. Those are tissues that are chemically fixed, and ongoing studies and clinical reports over the years revealed the intra and post-procedural complications. For example, there's a risk of migration because of this anchoring that may not be sufficient. This is not sutured to the patient. Now, there may be incomplete sealing that may generate paravalvular leakage. And this type of leakage during the diastolic phase of the flow back into the ventricle is characterized by very elevated shear stresses. Those are kind of microjet that may activate the platelets yet again. And because of that, in failing such procedures or after quite a few years of operation in the patients, we may have thrombus formation that may eventually fail the whole prosthesis, and there's increased risk of stroke. Now, another problem that starts to be more and more severe as we find out with follow-up studies is cardiac conduction abnormalities. When you deploy and anchor the TAVR, it pushes against the atrioventricular or the AV node of the heart, and the hoop stresses that are generated by the deployed stent of the valve may induce this cardiac conduction abnormalities that may require a certain percentage of the patient's implantation of a permanent pacemaker. Very problematic. Now, another thing is the durability of those valves. When we crimp them, and this is fixed tissue, we actually may damage the valves. And we need to crimp them to very small diameter. Femoral artery, clinically it's referred to in something that is called French, usually to the size of 14 French, which is less than 6 millimeters. So you really crimp the valve very hard, and eventually it may damage the tissue and impair its durability. Now, we conducted many simulations to see what's going on with these devices, and can we optimize both the procedure and the devices. So we start, and all those are patient-specific simulations. So we obtain computer tomography angiograms from patients at the Istanbul University Hospital pre-TAVR procedure. And then we go through reconstructing the whole geometry. Eventually, for example, you can see here the complex geometry of the calcified aortic valve before deployment. And we characterize it with advanced material models, also for the calcifications and for the soft tissues, etc. And we actually simulate the whole procedures. For example, the expansion of the valve is done either by self-expansion or by balloon expandable stents, like the case of the Sapien 3. And here, for example, you see the crimping stage which is simulated by us, and then together with the valve itself. And here you can see the correspondence between our in silico simulation and what we see in vivo. And you can see that it's pretty faithful to what we obtain in vivo. This is just for the procedure itself. Now, we did that in order, for example, to see how well the TAVR transcatheter aortic valve replacement anchors into place in patient-specific geometries in different locations. So here you can see the expansion in different actual locations. And we actually, that mimics all kinds of clinical practices, how you go about it without going too much into the details. Now, we wanted to see a case of pretty catastrophic accident that happened during one of these procedures at the Stanford University Hospital, in which after deployment, the valve migrated into the ventricle of the patient. The patient was eventually saved, but needed to undergo full cardiothoracic surgery. And apparently, the valve was deployed in a too proximal kind of position. And our simulation showed that it could have been avoided. What you see here is the green curve is the case of the actual deployment according to the actual location. And this is the total contact area. And you can see here total loss of the area. The two other locations, if it was deployed like that in that specific patient geometry with the presentation of the disease, it wouldn't have happened. So this could be used to inform the interventional cardiologist to perform the procedure. Now, in the last couple of years, we started collaborating with Simulia Living Heart Project. This is a big subsidiary of Dassault Systems, the actual airplane manufacturer, huge corporation. And this is a full electromechanical pumping heart that mimics the full cardiac cycle, because we wanted to see how such deployment would look in a pumping heart. And I'd like to show you some of these simulations that we conducted. This is the Evolut self-expanding transcatheter aortic valve. In a minute, you're going to see what happens to it when it's anchored into place and the heart actually starts pumping. You can see that it's moving around quite a bit. You can see the stresses on the leaflets, etc. But mostly we were interested both in anchoring contact force and contact area. And here you can see another such simulation of the Sapien such a device. And here we were very interested to estimate the risk of cardiac conduction abnormalities that may require implantation of a pacemaker. And in collaboration with Georgia Bossi from the University College of London, we studied many cases of such patients, and we've identified a cohort of patients for which the stresses next to the AV node, where the deployed valve pushed against the node, were double the average of all the other cases. And guess what? Those were exactly the patients that required the permanent pacemaker implantation. So this could be also used in order to analyze a specific patient geometry to see whether there's a risk of cardiac conduction abnormalities. Now, we also do the same thing about estimating according to the deployment location, the thrombogenic potential, and mostly during the backward flow or the diastolic phase, where we have elevated shear stresses that may increase the risk of thrombosis. So we're doing the same with the stress accumulation. Those are platelet trajectories flowing backwards through the deployed device, and we have the thrombogenic footprint, as you can see here. And in that, again, comparing different locations for the deployment, we identified certain deployment strategy that would minimize this thrombogenic potential. But of course, there's a lot of trade-off between the two, but you can see that all this in terms of planning the procedure and improving the clinical outcomes can be pre-planned with in silico simulations. Now, how well does it correspond to in vivo findings, for example, in patients? So here you can see echo Doppler and the location of the paravalvular leakage through the valve, this flow going backwards, that indeed the numerical simulation predicted very well, exactly the same location. And here you can also see a fluid structure interaction during the pumping cycle of flow through the device. You can see how complex the flow field is. So I've described actually a lot of problems and limitations of this tissue-based technology. So we set out to develop competing technology that is not tissue-based, and instead, and this is a startup company that we started at Stony Brook, also in collaboration with the Sadler Heart Center in Tucson, Arizona, of developing a polymeric valve that will have better hemodynamics and also longer durability. So in that, it also lower cost. In fact, you need to understand that those tissue-based valves are harvested from all kinds of tissues, but many of the specimens need to be discarded, and by using approximately 5% of those harvested. And then there's a lot of manual work of actually suturing the tissues into the stents, etc. So the polymeric valve that we've developed, one of the unique features is to optimize its design for reduced flexural stresses during the valve opening and closing. And this was based on numerical simulations and a design that offers tapering, varying thickness of the leaflets, something that you cannot do with harvested tissue valves. And this is based on numerical simulations. And for example, this is a bioprosthetic surgical valve, and the type of stresses that you see during the diastolic phase that expose the leaflets to the high stresses. And this is the polymeric valve. Also, crimping stresses for transcatheter aortic valve replacement. This is the Edward Sapien 1 crimped, and this is our polymeric valve crimped. So you see much lower stresses, which actually may enhance its durability. So we actually, after optimizing the design, we built prototypes. Here you can see results of testing according to FDA ISO-compatible left heart simulator that actually tests the valve. And you can see here the opening and closing compared to currently used surgical bioprosthetic valve and TAVR valves. Here you see fluid structure interaction modeling. This is in this idealized geometry of the left part simulator. And just a bit of secrets of our device, how we fabricate it. And this is a special hemocompatible polymer called Crosships that is in clinical use by Boston Scientific for paving stents and also for glaucoma patients. But we've collaborated with its inventor, Len Pinchot, for coming up with this specialized customized version for prosthetic heart valves. And here you can see that its thrombogenic potential measured with a platelet activity state assay is comparable and less than surgical bioprosthetic advanced life sciences valve and far less than another tissue TAVR valve. It's much more biostable. A huge problem of such valves is formation of calcifications. So this is a tissue valve after in vitro calcification, and this is our valve with hardly any calcifications under the same protocol. Crimping damage, usually in tissue valves, you don't crimp it beyond 60 minutes. So you just crimp it just before the procedure. And we tested our valve even after 8 days of crimping, and we can hardly see any damage as compared to tissue valve, which means that our polymeric valve could be delivered pre-crimped. Now, the one thing that we are most proud about is extended durability. In order to meet the FDA ISO standard, your device needs to be durable for at least 5 years in a patient. You do this by testing accelerated wearability in special devices. Our valve, what you see here is when it's reached 600 million cycles, but now it reached 800 million cycles, which are equivalent to 20 years inside the patient, which means that they will fit pretty well both low risk and also younger patients. Also, it is a much lower cost to fabricate them and more reproducible for mass production. We are currently going into in vivo studies in animals. Now, I just wonder how long do I still have for the presentation?
We're getting towards the end of the class time, but if you want to take a couple more minutes to wrap up, we can start questions.
Okay, so I'll very quickly go through something completely different, which is multi-scale modeling of thrombosis across the scale. So I will rush through, I will not go through all the interactions of platelets with injured endothelial wall, which is the inner lining of blood vessels. Here, there's a lot of biology involved. But we completely departed from continuum approaches and went to molecular dynamics and concentrated molecular dynamics and dissipated particle dynamics in order to model that, because we wanted to model what happens within the platelets. So here, both the blood flow is modeled by particles interacting with particles through energy potential and forces. And this is the internal structure of such a platelet. And the modeling is intended to also mimic the formation of these extensions of philopods that happen upon platelet activation. Now, next slide is actually, okay, I'll go straight to that because what's going under the hood is very complex. So imagine a platelet flowing in blood, Couette flow type of shear stress, we actually quantify, and the platelets is flowing and flipping at the same time, quantify the stress accumulation. Those are the red dots on the membrane of the platelets, and a certain kind of level. There's going to be a mechanotransductive process that will tell the philopods to grow. And here you can see what happens then. Those kind of filaments start growing and extending to the platelet membrane, and we end up with an activated platelet. And then this also influences the flow dynamics around it. Here, for example, you can see recruitment process of a platelet when another platelet already resides on the vascular wall with other receptors that are conferring this type of simulation. Here you can see the formation of a triplet of platelets in free shear flow, and you can see also the stresses that are induced on the platelets that eventually will also extend the pseudopods. But here we already see the attachment that forms the platelet aggregate. Now we conduct, of course, many experiments to validate this type of simulations that you can see right here, the flowing blood. We actually built a huge database of such cases. Here you see recruitment of flowing platelets to platelets that are already residing on the wall. So this is the initiation of thrombus on the vascular wall. And here, for example, you can see a model of a platelet semi-attached to the wall and flipping around. And here you can see how we measure it with video microscopy. So this is a high resolution, high-speed video microscopy. But as you can see, this is very sparse in vitro data. And the question is how we validate and compare directly the two methods, the simulations and the experiments. And in that, we're using machine learning. So I won't go too much into the details of that. I don't have enough time, but we harnessed machine learning both for identifying the shape of or irregular shape of activated platelets, and also for reducing the computation time that could be prohibitive to this very large scale simulations. This is some of the machine learning algorithms that we're using. And here, for example, you can see predictions of platelet flipping. On top here, you can see in vitro experiments on the vascular wall, the prediction of the machine learning. You can see right here, this flipping obeys certain frequency that is predicted by the simulation that we can also measure and validate experimentally. Here you can see identification of irregular shapes of platelet activation by machine learning approaches. And here, again, part of the methodology that enables to identify even the most irregular shapes that we may encounter and eventually predict by the models. And also, in order to cut the computational time, we in that also introduce two algorithms. One is called multiple time stepping, the other one adaptive time stepping, and eventually moved to something that is called artificial intelligence adaptive time stepping. And I'm really rushing through it. This is part of the architecture that enables that. And here, for example, you can see platelet flowing and flipping in a flow field and mapping the flow stresses and guaranteeing that this, which is kind of coarse graining of the computation, still predicts the same results with far less computation time. And here, for example, you can see the amount of computation time that is getting saved, that eventually what may have taken on some of the fastest clusters HPC resources in the world, like over 2 years. Eventually, we can perform this in approximately 12 days, and now even lower than that, using one of these special Amos IBM machine in White Plains, New York. So in that, I would like to thank the real talents that helped me create that, my biofluid research group members. This is from a couple of years ago. Many of the students here already graduated with a PhD. Some of them remained for postdocs. Some of them moved to academia or the industry. All my collaborators from the Department of Medicine, Surgery, Radiology, from the Stony Brook Heart Institute, the research support, and there were several, one from NIH, NIBRB, the Quantum Award, which is a very unique award. For the multiscale modeling, we got awards from the National Heart and Lung and Blood Institute, and the event for the polymeric valve, STTR phase 1 and phase 2 awards, and also another NBIB Bioengineering Research Partnership Award, and also some support from New York State. And as we can see, we use a huge cluster at Stony Brook University and the IBM Watson Research Center Amos cluster. And we have quite a few industry partners, including our own Polynova cardiovascular startup. So with that, I'll be happy to address any questions that you may have.
Awesome. Thank you so much, Danny. That was great. I always say the saddest part about virtual seminars is that we can't actually give you applause, and so we're limited to, you know, little clap reaction. But that was a really fabulous talk with a ton of just really cool data and
I appreciate it.
At this point, I'll open the floor for questions. If any of our faculty members want to unmute themselves and ask questions. I know Alan and Joel have signed on, Danny, so if you want to, you know.
Oh, hi, Alan and Joel. I wish we could have met in person. Have a nice, fresh and cold IPA.
Yeah, we were so looking forward to you being here in Birmingham, Danny.
And we could have jammed a little as well.
Yeah, of course, yes. We'll try for next year.
Yeah, absolutely.
Great talk, though. Thank you.
Thank you so much.
Danny, I'm sorry I missed your introduction, but I came in a few minutes late, but I hope MK explained to the crowd here that you are almost as good a guitar player as you are a scientist.
I was going to let you do that part. That's why you should have been here.
First of all, I want to tell everyone that I obey to the real czar here, Alan, who is like the leader of our Bedrock group. So kudos to Alan.
Good to see you, Danny. That was an awesome talk. I'll ask a science question, and you know I'm not a fluids guy, but your LVADs reminded me of turbines, and you always talk about shear stress, but you never mentioned cavitation. Is cavitation even possible in blood?
Absolutely. Actually, I started my research career studying cavitation in mechanical heart valves. And what we've revealed there is that actually it happened with leaflets of mechanical heart valve, where patients were rushed to emergency rooms with cracked leaflets, and when they examined the damage, it was a cavitation damage. And this was unheard of. Cavitation in blood. Well, you have during the last closure of the leaflet approaching the valve body, formation of microjets that I've studied, and in the core of these microjets, the pressure drops so fast that you have formation of cavitation bubbles, and that eventually induces cavitation damage that may eventually crack the pyrolytic carbon that the leaflets are made of. Ventricular assist devices may also do that, but to a less extent. But that's a different mechanism. I won't go into details about it, but yes.
Wow. All right. Thank you. Appreciate it, man. It's good to see you.
Any other questions? If our students have questions, please put them in the chat so I can moderate those. I do know we have other faculty members from across both BME and other departments that are logged on too. I look back through our participant window.
I would like just to add that if anyone have any question about that, wants to get more information, feel free to contact me via email or seminar.
Absolutely. And if you can't find his contact information should be in the announcement that went out, but I can also get students in touch with Danny if you guys want. I was going to ask a question about the calcific valve replacements.
Mm-hmm.
When the balloon actually expands and you basically push the calcific valve out of the way, how much does the kind of debris from the old diseased valve interfere with the ability for you to predict flow after the implantation?
Excellent question, because there are indeed the debris. Sometimes during the procedure, you need to use all kinds of filters, and you would like to remove them, or you need to dissolve them in a different manner. But I'd like also to draw the attention to another problem. Well, this one is mostly solved with kind of close supervision of the patient immediately post-op. Another problem is that sometimes those calcifications are really large, so the deployment eventually cannot totally push them, and you get an eccentric orifice. So now you have an impaired hydro or hemodynamic performance of such a valve, where, for example, one of the leaflet doesn't fully open or close and stuff like that. We are now working on eccentric designs for TAVR valves, and also specifically for bicuspid aortic valve patients, for whom right now there's kind of off-label deployment of regular tri-leaflet type of TAVR valve, which is far from being an ideal solution for them.
Okay. So thank you. Do we have any questions from anyone else in our chat room? I'm not seeing any, but well, I think that's it for the questions, but thank you again, Danny. We are so glad you could come virtually, and we're very sad you couldn't come in person. We really appreciate you taking the time out of your day to present this us. I also have to tell Alan and Joel, since they missed the intro, Danny won the Salvio Wu medal for ASME. So he's a rock star again, both at the bedrock level and in the science lab.
Just I wanted to mention that I was notified about it yesterday night by the director of the ASME. So this is the Savio Wu kind of a translational bioengineering award.
Wow. Congratulations, Danny. That's awesome. For those of you who are not in ASME, this is a really, really high achievement award. So congratulations.
Thanks so much,
Alan. So with that, I'll conclude the main body of this seminar. Danny, if you'll stay on for just a few minutes to make sure we can take care of any administrative details with Carrie and our students can log off. I will see our students next week for our honors undergraduate presentations on Friday, December 4th.
Okay.
Not next week.
So I'll stop sharing right now.
Yeah.